My name is Mike Griswold. The film that follows gives a detailed description of the methods we use here at Washington State University to culture primary Sertoli cells from the rat testis. I was fortunate to be in the laboratory of Dr. Irving Fritz when Jennifer Dorrington worked out the basic methods that we use. A number of important modifications of this procedure have been developed by Alice Carl, and she will demonstrate them during the course of the film. At least two other methods developed by Wiebe and Welsh and Anna Steinberger's laboratory are currently in use. All of these techniques allowed for the first experiments on the biochemical responses of Sertoli cells isolated from other testicular cell types. A number of important discoveries have been made using these culture methods, including the response of Sertoli cells to FSH, the characterization of secreted proteins, and the action of growth factors. It is important to note that these procedures allow for experiments on Sertoli cells from rats between 10 and 35 days of age, and the cell cultures are done in minimal medium in the absence of added serum. Results obtained from cultured cells should be verified with experiments in vivo. Recently, some Sertoli cell lines have been developed using the SV40 virus. These cell lines appear to retain many of the characteristics of primary Sertoli cells and may be useful in future experiments. The slide you are now viewing came from the laboratory of Irving Fritz and was put together by Dr. Pierre Tung. It visualizes the process of putting Sertoli cells in culture utilizing the scanning electron microscope. In frame A is a picture of the seminiferous tubules and including the interstitium before any treatment. In frame B is a close-up of the seminiferous tubule before treatment with trypsin, and in C is a lower power view after treatment with trypsin, and D a high power view showing the layer of collagen and peritubular cells surrounding the tubules. Frame E shows the tubules after treatment with collagenase, and frame F shows the fragments of tubule just before they are plated into the culture dish, and these fragments contain both Sertoli cells and germinal cells.
All right, let's get underway with this process. And let me point out at this time when we start that until we get our material into the hood, we will be carrying out only a clean process. I have killed our animals here by cervical dislocation. I prepared ahead of time a small petri dish with hanks in it that's properly balanced, pH balanced. And we take our animals and arrange them so that they're conveniently set up for us. I like mine with the tail away from me, and then spray the body with alcohol. And this is done just to wet the fur so that it won't stick to your instruments and get into your tissue collection. Notice that your instruments are kept in separate containers. You have one set of instruments to open your animals, and you have one set of instruments then to remove the testis with. And that's an important thing to do, because otherwise you will wind up with hair inside your material. Let me turn my hand around. If you lift the skin and make a cut in this manner, clear through all layers of tissue, and then take the rounded tip of your scissors and introduce it into the opening. Keep it well up against the skin and push it down toward the leg. With one cut, you can get the entire abdominal flap cut and peeled back. It's very important that you not perforate the gut. If you do, you will have E. coli in your material and you won't be able to work with it. If you think you've perforated the gut, just dispose of that animal and start again with cleaned instruments. Your instruments, by the way, are kept in a solution of 70% ethanol. And of course, they don't remain sterile once you've cut into the first animal, but all you're really doing is a clean process here. I always open all of my animals at one time, just so I don't have to keep moving back and forth between the instruments. It saves time if you just do it as a group. I've finished now opening all of the abdominals cavities of these animals, and I've taken out a new set of instruments, and I'm going to use these to remove the testis from the body and place them into our little petri dish that we have here with Hanks in it, ready to receive it. And this Hanks is designed just to keep the tissue moist while it's sitting there waiting on us. And you just want to take the testis, pick it up by the vase, trim away as much of the fat as you can, but don't spend a lot of time at it. Your point is to get it into the dish as promptly as possible, not to put a lot of excess tissue in there because it will change the pH of your Hanks, and you don't want it to change on you too fast. Using the same instruments that you use to remove the testis from the animal, arrange your testis out on a piece of parafilm. Hold it lightly at the end or on the fat pad that you have remaining there, and just snip into it. Take the side of your scissors, flat side, lay it along the testis, and just push the tubules right out. And as you do, you'll see this major vein that comes with it, and I usually just take the same side of my scissors and lay it down across that vein, pick up the tubule bundle with my tweezers, and pull gently, and it will tease out most of that vein. And take your tubule bundle and drop it into your second container of Hanks. You will do each one of the testis in this fashion. Don't spend an awful lot of time teasing out the vein, but get as much of it as you can. All right, now we have removed the tunica from all of the testis. We're going to tip the dish a little bit and remove the bulk of the solution that's in there. Don't worry about drying them out entirely. This is an approximate weight that you're getting that will just allow you to figure out solution quantities, that's all. And you can see that I have already taken the lid of this dish and placed it on the weight and tarot it. And I'm just going to dump the material from the bottom dish into the top. Try not to pour it out onto your scale like I just did. All right, get your weight. Place the bottom over your tissue. Take it away, you're ready to go. Our tissues are all prepared, ready to chop now. Dump them onto your chopping block, and by the way, this chopping block should have had time to air dry, or you should have dried it from its ethanol wash before you dump the tissue on it. You don't want a lot of ethanol on your tissues. And just take a razor blade, chop down through it, sort of generally until it loses form is the best way I think I can describe it right now. You really can't chop it in exact pieces in spite of what we often say in recipes, because after all it's not oriented in such a way that you can do that. Okay, now you're ready to put it into your trypsin enzyme solution, which I have prepared ahead of time. And the pH is already balanced. So just lift your block up, or whatever you're chopping on, and scoot it off into your flask. Keep your fingers away from the edge that you're going to push your tissue across. Remember, you've become sterile since you've entered this hood environment. Recap your flask. Shake your tissue quite briskly. And now you're ready to put it into the water bath for the digestion process. While our material is in the trypsin digest, I'd just like to make a point about the Hanks. The importance of it being a calcium magnesium free solution What that does for you is it dilutes the ions off of the surface of the tissue so that they don't aggregate together as tightly as they normally would. It assists in separating the tissue. If you have ions in your solutions, you'll have to sort of work against that, and it makes your job more difficult. I'd also like for you to pay attention to balancing the pH of the hanks, because it's important that the pH not be too far off from the 7.4, 7.5 that you're looking for routinely. You can see I have two containers here. This one, of course, is not yet balanced at all, and it's quite acidic. This one is at the proper pH. A quick way of checking the pH that I find works really well for me on hanks is to just take the bottle and shake it. And as you see the bubbles come up, if they have any sort of blue cast to them, that will tell you immediately that it's a bit too alkaline and that you need to add a little more unbalanced hanks or something to bring your pH back in line. I prefer balancing with just more hanks in order to just sort of keep my solutions even. We've been checking this material every few minutes during the trypsin digest, swirling it to see how well it's coming apart, and we're going to check it now to see how well our DNase is working in this solution. Be sure when you get ready to take your flask from the water bath back into the hood that you wipe all of the moisture off of the flask. Take an aliquot of your material up, and I have deliberately put less DNase than I knew would be necessary in this so that you could see this really well. And if you see this sticky solution that's coming out, that's representative of a lot of DNA. That has to be broken down, otherwise your tubules will get tangled up in it and your cells won't come apart correctly. When you get the correct amount of DNase in your solution, you should be able to drop this material back off of the pipette drop-wise without seeing any stringiness whatsoever. DNase, we have found, is quite labile. It's reasonably labile in the powder form. It's very labile once you put it into solution. So we always make ours up just as we're using it. And it's pretty acidic as a rule, but you're adding it in such small amounts that routinely it won't really change the pH of your solution. And what I've done here is added another aliquot, and I'm going to go back into the water bath with this incubation, and I'll be checking it again every couple of minutes during this last 10 minutes of the digestion process. Now we've added more DNase and we've monitored this solution during the last 10 minutes of the digestion process, checking it closely. Once again, we're removing it from our water bath and wiping the water from the flask, moving it into the hood before we take the top off. Take a look at your solution at this time, swirl it, and you should see separate tubules primarily. There should be very little clumping, if any. And the solution should look just hazy as the tubules swirl through it. At this point, we're going to set it down and let it go through the first settling step. As soon as the tubules have collected at the bottom, then we will take off the majority of the hank solution that's on it and add trypsin inhibitor to stop the action of the trypsin digest. Remember that there should be absolutely no viscosity in this solution. You shouldn't see any stringiness at all. You can still add DNase at this point if there is some problem with that. If you have clumps, it's better to go ahead and digest just a little bit longer, but of course you can go too far with this digestion, so it's really a judgment call each time. But things should be well separated at this point. Our solution now has settled. If you tip the flask over, you can see the tubules gathered very separately at the bottom. And we're just going to pull off the majority of the solution that's on them, so that our trypsin inhibitor that we add to the flask can get right to the material and go to work. Be careful when you do this step. Don't suck your tissue up. Sometimes it tends to stream up. I like to take this down just as far as I feel like I safely can. Okay, we'll stop there. This trypsin inhibitor solution needs to set on this material for at least a minute. And then we'll add some more hanks and swirl it around to wash it. We do that process of washing 2 more times, and then we go into our collagenase digestion step. We are in the final washing step after the trypsin digest. Note that the solution at this point, after your tubules have settled, is quite clear, and this is how it should appear. We'll take off the majority of this supernatant also, and we have made up our collagenase digest solution while we were waiting for these settling steps to occur. We'll bring our tubule mixture up to the right volume, and we'll add it to the collagenase digestion, and then go through that digestion process. What I'm doing here is just pouring it into a tube so that I can measure it. One way not to lose a lot of material is just take a little bit of hanks that will be a part of what you're going to suspend your tubules in, and wash it around in your flask. If you're working with very few animals, it's really important to recover all of the material every step. Okay, your collagenase digestion solution has been made up, and you have a total volume of 15 mils, so you've brought your tubule mix up to 15 mils, and you're going to add it to the digestion flask. Cover it, and we're ready to go back into the water bath for that process. Now, it's really important to monitor your tissue carefully during this digestion process. It will digest very rapidly in the last few minutes, and you don't want to let it over digest, or you will not be able to do the separation that you get on the slow spins. While we don't routinely look at aliquots of the material after the trypsinization process, if you were to look at it on the microscope, you would see that the tubules have a very sharp demarcation along the edge. It might appear to you as sort of a dark line. You will see a few germinal cells in the field. They're just floating out the ends of the tubules, and you will see removal of the interstitial cells floating through this material. We have put our tissue through the collagenase digestion process. We've checked it frequently. We've monitored for DNase, just as we did through the trypsin process. Now we've taken an aliquot of it and put it onto a microscope slide and float a cover slip on top of it. Be sure that it's got enough solution in there that your tubules will float separately from each other, and then monitor the release of the peritubular cells from the surface of the tubule. Here we see an aliquot of the material taken from the collagenase digestion process about midway through. You can see that the edge of the tubules is beginning to break down. The collagenase barrier is dissolving away. Though there is no calcium and magnesium in the hanks, there's adequate calcium on the surface of the tissue for the collagenase activity. At the end of the collagenase digestion, you will see that the tubule has a very rough surface. At this time, the collagen layer is entirely gone and the myoid cells have been released, and you will see them adhering to the glass slide. If you go just a bit further in the digestion process, you will see that the tubules will begin to break apart, and you will see pieces of the wall of the tubule have broken out. That's what we see in this field at this time. We have completed the centrifugation steps that come after the collagenase digestion process. Here is our material after the final spin down. You can see that the supernatant is quite clear, even though it is designed to hold single cells. They should basically be gone by now, and this final supernatant should be pretty clear. I'm going to remove this supernatant, and then we will suspend our cell pellet in hanks. I use hanks in preference to medium because you still have the same ion-free solution that keeps your material from wanting to adhere to one another. It keeps it dispersed better for plating. After I have added the proper amount of solution to bring my plating mixture up to the volume called for in our directions, I'll pour the entire thing into an Erlenmeyer flask. I like to put mine into an Erlenmeyer flask because it gives you a broad base to swirl the material around in, and that's almost impossible in a tube of this kind. When you resuspend your tubules at this point, you should do it just by inverting the tube. Don't shake it hard. You don't want to break things apart at this point. And particularly if you have digested a bit far, sometimes your tissue is very fragile. You should have a nice even suspension. You should be able to swirl it. I do mine in threes. I pull it up like that. If you do it in a big pipette like I do, you need to sort of keep it laying sideways so your tubules don't drop out of solution. Lift your lid off. Shoot your material in. It's not a good idea to use a pipette man for this process. The shear pressure that's applied to the tissue as it passes through the small orifice that's used on those pipette tips generally breaks your material a whole lot. And there you are. In this dish of freshly plated tubules, you'll see that the tubules are still floating free in the medium. They need to be placed in the incubator and left undisturbed over the next 48 hours. They will attach to the dish and the Sertoli cells will migrate out from the edges. In this 10-day-old culture, the Sertoli cells have spread to form a single layer. However, don't expect this to form a continuous monolayer. Since Sertoli cells do not divide in culture, this will not occur. Sertoli cells are easily identified by the lipid droplet inclusions, which increase over time in culture.